our next speaker is Jose Napolitano, and he is going to talk about benchtop NMR to support small molecule pharmaceutical process development. So
Thank you. Thank you, Dave, and thank you, everybody. Thank you to all the organizers for the invitation. It's just great to be here. Actually, my first ENC, so it's great to be here. Thanks also, Eric, for connecting us with the outside world. Appreciate it. So today, what I would like to do is to tell you a little bit about our efforts to bring benchtop NMR outside our lab and use it to support a process development on a pharmaceutical environment. So the first thing, just a quick note on pharmaceutical process development is a little different from what we see in discovery, which is very, very valuable. And, NMR has a very big role in there. For process development, we kind of already have a candidate, right? So what do we do with it is very important. And at this stage, what is very important for an organization like the one that I'm part of is to make sure that we have a sound, efficient, synthetic route to meet supply requirements. So, when we start, we're talking about a few milligrams for discovery, which always makes the life of our NMR folks very difficult. But as you move forward and you're going into clinical trials, phase 1, phase 2, the amounts will increase. And so we have whole teams of synthetic chemists and analytical chemists working on these projects. NMR is obviously an important part of it. And high-field NMR has been a mainstay technique for process development for a really long time. I think for an audience like this one, I don't have to make much of a case that those instruments are not portable or movable. And so there are a lot of applications in which we would like to have the NMR closer to other instances, other facilities. And the benchtop NMR offer that possibility. Of course, not everything is perfect. There's going to be some penalties in terms of sensitivity or resolution, but I hope that with today's presentation, I can show you some ways that we're trying to attack this problem. It's going to be a lot of how we learning opportunities for us when. So I have two specific cases that I want to go through today. The first one is how our attempts to bring NMR to a chemical warehouse and just apply for rapid ID. And the second one connects a little bit with what Manuel shows at the end of his presentation, that is bringing the NMR process closer to our process chemists and talk about process development and optimization. So for the first one, chemical warehouses are fascinating to me. They hold crazy amounts of material, and you will think that mix-ups don't happen, but they actually do from time to time. And yeah, a drum of acetone and a drum of ethyl acetate look very similar. And if you don't have the control in place, you can really mess it up. So our main goal here was to try to use the NMR just for rapid ID testing of very small molecules, solvents, acids, base. These materials are usually held in the warehouse, and they have to be moved somewhere. You have to sample the drums and move them to a site facility when they are tested. Commonly Raman, you have IR, you have GCFID. So we were thinking, how about having the NMR closer to the warehouse? And this was a really, really fascinating collaboration with another company, with AbbVie. So we have two pharmaceutical companies able to work together. Obviously, we're working with solvents, so proprietary information is not kind of an issue. Also using two different instruments from different vendors. And so one of the main challenges that we thought about at the beginning is who's going to take care of this? Who's responsible? Because obviously the expertise declines very quickly as you move away from the NMR. But training, automation tools, reliance on software, making sure that your standard operation procedures are very crystal clear are important here. So let me give you a few examples of how we try to tackle these things using the NMR. And again, you will see kind of a progression on our learning and also some of the limitations that we encounter, or the challenges that we encounter along the way. So the first thing that is clear to us was that, if you have access to carbon-13, well, you are golden. Carbon-13 for the win on this one. There you see just a quick comparison between a high field and low field experiment. And this is a very, very, well-established workflow in which if you have clean signals that you can easily pick, you can quickly go to a database and get a match. This example shows a carbon, but work with depths, it can work with HSQCs. So this is a kind of our main approach, but not everybody has carbon. So what do you do if you don't have it? So resolution starts to be an issue, and then it really depends. For some of these very small molecules, you have little to no signal overlap. It's easy to pick. It's easy to integrate. So you can use a similar approach. Not so much of a problem. But these molecules don't have to get much bigger to start getting very complicated. And then now, things that are very common to us, like integration or picking, start being difficult. And even if you decide that you are going to try to do a visual inspection, even those are not very clear to do. So what do we do in these cases? We try to approach it in different ways. I will show you in the next few slides, but first I couldn't resist to show a spectrum of ethanol, which I think in an NMR conference is kind of criminal. But there are definitely different types of ethanol out there. This is denatured ethanol that can be used as solvent as a process, for example. And you see that in this case, we have two different grades or types, 2B and 3A. Both are ethanol, but you certainly don't want to use ethanol that has methanol as an excipient if you are doing, if you're running into the possibility of having an esterification reaction, a side reaction. So these materials, this kind of ethanol is commonly released by Raman, which I think that is pretty interesting because I'm pretty sure that ethanol and butanol and most of them are going to have similar responses. So NMR provides a very easy way to look into these materials. In this case, not only verifying that it's actually ethanol, but also looking at some of the other components in there, like toluene or methanol. So going back to complexity on the signals, we have been working with different softwares trying to understand what is the best approach. And to be honest, it's not there's not like a gold standard or an all size, something that fits all solution. We have been a lot of work with quantum mechanical spectral analysis. In this case, in collaboration with Matthias Niemitz and the people at Cosmic Truth in Kuopio, Finland. And you can get exquisite profiles for some of these small molecules. There are caveats, of course. Obviously, these small molecules don't have a conformational, you commonly don't run into conformational problems. But you can get very reproducible patterns that you can then use for a new sample. The advantage also from a purely structural chemistry guy is that you get full assignments, chemical shifts. If you want to do, thinking about something like chemical shift prediction, you can get very, very accurate values, coupling constants, relative integration. So it's a very nice application if you want to think about the structure, right? That might not be necessarily the way that you want to go. You might want to use some chemometric approach in which you're not necessarily thinking about the structure itself, but just how the overall pattern on the spectrum is different. So in this case, we were using classification using PIXAT, software from SPAC, and we can get a very minimal misclassification. In this case, I think I have like different 30 solvents, but we have gone up to 50. And this has been very helpful to go over our usual data interpretation procedure as an NMR spectroscopist. Related to this chemometric approach, I have an example that is kind of interesting, is that all these samples were analyzed neat. There is no deuterated solvent. So you have to come up with strategies to reference the spectrum for each individual solvent. And then there is these ones that are very interesting, right? Like what happens when you have a single signal. Is that, well, integration is not going to help you because it's not relative. You don't have any couplings. You don't have any really chemical shift because you are setting up the reference, right? And we run out into a very interesting situation that if you run neat acetone and neat DMSO, they cannot come up exactly at the same place. So, okay, as an spectroscopist, you can, zoom in and look at coupling constants and say like, yeah, now I know based on the heteronuclear coupling which one is which. But I think that using the chemometric approach, actually, you can clearly differentiate them. And in this case, on this small graph that you see there, actually, if I on purpose misclassify an acetone sample as DMSO, the chemometrics approach will be able to get it. So for the second part of this presentation, I would like to talk a little bit about how we're trying to bring this instrumentation also to the process chemistry lab. And again, the main idea is to support process development and optimization, understand how the reaction works, but also extract concentrations, kinetic information. And here, the main challenge that we always encounter is avoid that urge to adapt the process to the NMR system. So what we're trying to do is make sure that the process chemists can run the reaction of interest as close as possible to the actual conditions that are interested on. So avoiding deuterated solvents is a clear example of making sure that the concentrations are the ones that are really interested in. Before going into reaction monitoring itself, I wanted to show an example that I think is very interesting. It's a solvent swap. So sometimes we have just a solution. We have a compound of interest in solution, and we have to put it into a different solvent. And you might not be able to completely dry it out or completely isolate it. You just want to telescope it into the next step. And for that, you will need to change the solvent. And so what you see here is a very simple operation, just a simple proton collected by one of our process chemists. And using craft, we can just get the ratio between NTB and heptane. And that way they know how to continue. Of course, as they are producing new materials, they are also interested on doing a quick ID on the fly. And so this starts to look more like the molecules of our process chemistry commonly sees. And what you see on the screen is the proton spectrum on our 60. And so the question that we get from them always is, does this pattern make sense at all? Excuse me. And so, again, using QMSA in this case, we have developed methodologies in which we can create a profile at high field, because we are always going to have some data at high field, and then apply that profile at the low field. And so over here, what you see is just the models of every signal on that structure. Quantitation is also very important for us. This is just a variation of the slide that Manuel just showed you. Of course, we are very interested in having access to concentrations without worrying about relative response. That over there looks on the slide looks like an ordinary calibration curve, but it's actually the results of having 4 different standards to calibrate our samples. And so this has been also very beneficial coming to reaction monitoring and understanding what are the concentrations of reagents in solution. This is kind of our test system for when we're starting to develop things. It's just to point out that, yeah, sometimes you have to deal with big signals, with a high dynamic range. Obviously now with the solvent suppression strategies, this will be something of the past. But This example really embodies what we're trying to look at. Get concentrations of our starting materials, any reagents that we're adding, when the reaction is really completed, are there any intermediates that we want to track? And I have a couple of examples that I want to also share with you. The first one is the formation of a Grignard reagent that are not exactly reagents that are going to be very stable if you are not careful. And for our chemists, the important part is to produce this reagent and understand how long you can hold it before you put it on to the next step. And so what you see here is just a long-term stability study to see how fast we can produce that Grignard reagent and then how long it takes to start the proto-demethylation process to get this degradant product. Again, in this case, you see the data both collected with hydrogen and fluorine, which is another important capability for us. One last example before I jump to the conclusions. In this case, using fluorine NMR, we were interested in understanding this particular activation reaction. And if you have worked with CDI before, you know that it's a pain. CDI degrades easily, microscopic. It's not only that bottles on the same lot can have different concentrations. It's that the top of your bottle and the bottom of the bottle have a different concentration of CDI. So it's very hard for the chemist to know what to charge, how much CDI to put in there. So in this little graph on screen, what you see is the first CDI addition. The reaction is pretty much instantaneous to produce the activated acid. But you can see that there are several additions of CDI that have to happen in order to get what you really needed. And then once you have reached the concentration that you are aiming for, after addition of the fluoroaniline, you can monitor the acylation product. All right, I hope that during this presentation, I have shown you how we envision moving some of these systems away from the NMR. There are certainly, there have been wonderful collaborations over the last couple of years. Still there? All right. You see that there are efficient workflows that can be used for ID and for reaction monitoring as well. And that software solutions are going to be playing a very prominent role as we move forward into more complicated cases. Again, this work is not possible with a very engaged team and very fantastic scientists and individuals, the SCG, our collaborators in ADVI, our process analytical group, Magritech for their support, collaborations with UBC and Jason Hine, and finally also all the people from our software companies that have helped us over the years. With that, I thank you all for your attention, and I'll be happy to answer any questions that you may have.
So, any questions? May I ask about how many systems do you maintain? Benchtop systems in your total.
So right now we have one that is our POC system. What we envision, and that's a conversation that we have had in the past, that we should have at least one of these on every process chemistry lab, at least one. If you can have one in every bench, would be better. But at this point, at the stage that we are, we have our POC system.
And for the solvent, do you have like a system in the warehouse?
Yes, so that system was moved during the POC, and what we're doing also is now understanding what is required for, let's say, GLP and data integrity purposes to make sure that is done in the final location. And the status on our company and on ADVI is different because of the requirements, but they are also pursuing the same thing.
Okay, thank you. Other questions? Okay, well, thank you.
Thanks.